So now let's return to the topic of T cells. This is what the cytotoxic T cell actually has for a weapon. It has little granules. You got to keep this nasty purple stuff away from everything else. And so the T cell's on top in blue and green. Bottom is the infected cell that it's destroying. And you can see, again, we have perforin. This is a perforating protein, okay? It basically pokes holes in the membrane. And it also lets some toxic proteins into the cell, but it also causes osmotic disruption just by itself. And then there's other things you can't see. There's reactive oxygen species. And as you know, oxygen is kind of important to me from reading the book. Reactive oxygen species are a big part of immunity as well as evolution. So yeah, this is what you have in here. You have granzymes, which are enzymes that actually break down the other cell, and the reactive oxygen species. And that's shown in purple, and you see how that is causing problems for the yellow cell at the bottom. In fact, we even see these, it looks like these little, some of these modules not only can go across an immune synapse from one cell to another when you're right there, it looks like these can survive a little bit in your bloodstream and maybe be long-range attack weapons. So that's what this is about. This is about a picture of a cytotoxic multi-protein complexes they call supramolecular attack particles, or SMAPs. And what a SMAP looks like, they propose that these are autonomous extracellular killing entities that deliver cytotoxic cargo from a distance. It's like the cell thinks it got away, right? But they throw a SMAP at it and the SMAP gets it. It's that kind of thing. So you have the SMAPs actually interact with integrins and other cell surface molecules on the infected cells. And this is what it looks like. They sort of literally almost looks like a virus, but this is a virus that's fighting for us. And it doesn't have genetic material, it just has all the nasty stuff, which in a weird coincidence is also shown as purple here. But you have the thing is, these are cool little assemblies of lipids and proteins that can actually they can survive in the body for a distance. So that's really neat. That's kind of how T cells work in a cytotoxic function. Now, the question is, what's going on with the other type of T cells? Because that was just a cytotoxic kind. This is the kind of T cells that I show as a radio. There's the helper cells, which are disarmed, but they do help the other parts of the immune system find the bad guy. Okay? So they're actually based on macrophages are an important part of this. Remember, macrophages, after they eat something, what do they do with it? Well, they break it up just like we do for all our proteins, but they these broken up proteins are from the outside of the cell. And so it puts them in a different kind of MHC called MHC class 2. And you see right here, it looks very similar to MHC class 1. It holds a little red peptide, goes to the surface of the cell, binds T cells. But this binds T helper cells rather than cytotoxic T cells. And look at what happens if you have a B cell and you have an antibody that binds an antigen. It gets internalized. It gets chopped up. It gets put on MHC class 2. So the same system is used by B cells as well. These are cells that deliberately internalize external proteins. But this is one of the ways that the immune system is used to fight off against those external proteins. And this is a crucial step in deciding that you should make more of this antibody. Basically, the B cell doesn't have permission to activate until a T cell comes along and sees this and says, hey, we've got to, I agree, this is bad news. Make more of these antibodies, start to clonally proliferate, and then everything else goes on. So T cells are how antibodies are made. They're actually deeply involved. And here's an example, like on a macrophage, you have the T cell that activates the macrophage, and then the macrophage starts to send out signaling molecules. The helper cell might send out its own signaling molecules, and those will recruit other parts of the immune system that actually have nasty things to attack the the bad stuff with. And this is a B cell. It will activate when it recognizes the MHC class 2, but not the peptide inside it. That's when it will activate the B cell, and you'll start to get proliferation. So remember, the whole key, these are acting like radios. These are not, these don't have cytotoxic particles in it, but they call the cells that do. So, I think I wanted you to, but this got messed up. Well, I think I have it, so I want to ask you to just make some observations here. How can we describe the T-cell receptor? You know, you can say how many domains, how are they organized into chains, and we've already established that it has antibody domains, but how are those domains organized? Just interpret this figure. Basically, how many domains do you see? See 4, right? How are the 4 domains organized into chains? 4 domains in 2 chains, but also you have a transmembrane region down here. And the transmembrane region, by the way, has these positive charges on it, which are important to how it signals to the inside of the cell. So there's part of it that goes into the inside of the cell. It looks like an antibody from the neck up, but from the neck down, it's actually a membrane protein. And it has an intracellular region. So that's the important thing, that it's membrane bound, and it has, and it has variable and constant regions, just like an antibody. These are regions, not full fragments, because you can't really fragment this very well. It's got a little bit of sugars, because remember, everything outside the cell likes to have sugars on it, and these are no exception, but you can study it without having the sugars on it at all. So if you look at the variable region, you would expect it to look just like an antibody. And in fact, it does. If I show you, it's not a fair question for me to show you this on a test and to ask you, is this a T-cell receptor or an antibody fragment? Because you can't tell. I can't tell. Maybe people who work on it can tell, but it doesn't matter. Each, if you look at the individual domain, you have, this is sort of a side view of the domain, and you can see the constant region right here. This is a end-on view of the variable parts. And you see 1, 2, 3, 1, 2, 3 equals 6 loops altogether. And they even look like antibody loops. We talked about how there was one that was longer than the others called CDR3. They have the equivalent, and they also label it loop 3 because let's try to keep things simple. Immunology is complex enough. So the most important loop, the one in the middle, and the one that's most likely to contact peptide, is the same as the most prominent loop in antibodies. There's some real similarities going on here. So usually the first 2 loops, L1 and L2 and H1 and H2, those bind the MHC. And then usually the loop 3 binds the peptide in the middle. Because you have to imagine that the MHC is a big protein, the peptide's small. It's only about 10 to 20 residues long, depending on the type. In fact, here it is. So what I think I even got this into my dissertation. I talked about it. The structure of an MHC is like a hot dog and a hot dog bun. This is like the hot dog where you're just kind of annoyed because the bun's too big for the hot dog. You're like, I'm getting a bunch of bread at either end. But that's exactly what MHC class 1 is like. It binds short peptides. And you see that because there's only part of the peptide that's sticking up. The rest of the protein is like a clamp around the peptide. And just one thing to note, I call these MHC proteins, they're also called HLA. Technically, HLA is a name for the gene that produces them, whereas MHC is the protein itself, or it can also be used for the huge gene complex. Anyways, basically, as far as I'm concerned, use them interconvertibly. But understand if somebody says HLA, it means MHC. It means this system. And so this is a window into the cell, but structurally speaking, at a protein level, it looks like a hot dog. If you look at the side of it, here's an MHC class 1 from the side, and you can see it's got, once again, we see IG domains down here at the bottom. And up here we see something that's unlike anything we've seen before. That's because its function is to bind peptides. We're looking from the side, so you can imagine the peptides like right here. MHC class 2 looks slightly different. It has a different domain structure. We're not focusing on that much this year. But just to let you know, it binds peptides. It's like the foot-long hot dog bun, because the peptide can hang out either end. And so the peptides for MHC class 2 are going to be longer. Peptides for MHC class 1 are pretty much constrained to be 9 or 10 residues. But peptides for MHC class 2, they're the kind where they're hanging out the end, and you can have 15, 16, 20. Doesn't really matter, because what matters is what's in the middle. That's where, oh yeah, I showed you peptide. There you go. So just remember that the T cells are like antibodies in that you have 6 loops contacting, and you see right here, the 3 loops, the loop number 3 is right in the middle. That's the one that's touching the peptide. That's the one that changes the most when you're seeing something new. All the other ones are around the outside, and so those are more important so that your own T cells recognize and bind to your own MHCs. And that's part of what the thymus is there for, is for us to select T cells that bind your MHCs, and they have loop 1 and loop 2 with the correct configuration. But this does mean that T cell therapy generally is a selfish affair, because your own T cells are used to your own MHCs. It's very tricky to take T cells out of one person and put them into another, because they have different MHCs, even if you're related. If you're related, you might have half the MHCs the same. But it's one of the big problems, one of the big reasons why T cell therapy takes longer and is more of a problem. But you definitely see loop 3 changing all the time, because that's the one, you see, it's touching the peptide. If there's a difference on that peptide that makes it something it's never seen before, if there's a cancer mutation that's right here, if we're lucky enough to have a T cell that responds to it, your T cells can respond to mutated peptides because they've never seen it before. They freak out. If, however, the mutation is down into the protein, it's like 2 over, and it's down into the protein, it's possible the T cell will never tell, because it doesn't actually touch those side chains. Realize that the T cell is only touching a few side chains, and that's what it bases everything on. So, let me ask you to just sort of hypothesize. We don't have a T cell receptor. Wells didn't run a T cell receptor version of the antibody. It's not the T cell bot, it's the antibody. Why would he do that? Given what T cells can do, and given that T cell therapy is good, why would Wells, especially with the background that you know of, why would he make an antibot but not a T cell bot? Any ideas?
It's expensive.
Why would it be expensive?
Well, aren't antibodies easier to produce?
And why are antibodies easier to produce? Because they're proteins, right?
Yeah.
Yeah, you have a whole protein production. I don't like making cells. I like making proteins, and so that's what I do. So I totally understand that because antibodies are proteins, it's easier to make proteins than to make whole cells. And definitely, I don't think you could have a robot that would do all the complexities of cell growth. I know that there are some students who go over to the Hutch, but it's one of the big tech jobs that you can do. And that's why biology classes are helpful, because I help you make proteins. They help you take care of cells. But to each of them, you know, you can learn from one to the other. Yeah, any other ideas or comments about why he did not do the T cell bot?
I was just thinking antibodies are also humoral, so like similar ideas.
Right. They don't have a transmembrane region. And it's, yeah, whatever way you can find to basically say that. Even though antibodies are bigger, remember antibodies are 3 times the size of T cells, but still, they're made to be soluble. You don't have to deal with the membrane. You don't have to deal with keeping the cell alive. And that's really the kind of thing, yeah, that gets to, if you know it, it seems too simple to be the full answer. But it actually is the full answer, you know? It's that kind of thing. So, there were a few places that talked about T cells, but most people were talking about antibodies because we're always in a hurry, right? Well, we were in a hurry to get rid of COVID, so we definitely focused on that, but there were a few companies, even one I had a friend who works for, that actually will sequence your T-cell receptors and tell you if you have T-cell receptors that would recognize COVID. I would have loved to get my hands on that data set, but it's a lot of data and it takes a lot of time to get. So T-cell immunity does take longer to kick in, but it has a broader base because what you're talking about is you're talking about recognizing all these little peptides. And they sample, if you have a coronavirus, there's like, I think I even added up the number. There's like hundreds of potential peptides. Your actual immune system must produce dozens of those peptides, and so you eventually learn to recognize dozens of different peptides. And so I was always worried about variants, but I wasn't as worried because I knew from the rate of mutation, you get mutations here and there. But if I have a response to 12 different peptides, and each of those is 10 residues long, maybe with only half of them pointing out, that's still 5 times 12, 60 peptides. To be able to get rid of all those 12 responses, you need the right mutation at the right place for 12 different ways. And so there's a lot of redundancy in the T-cell system. Once you get it set up, it will provide a nice wall. And so I wasn't as worried. Oh yeah, and right here I have. Typically, what you are looking at after you've been infected with COVID, your T cells remember 15 different fragments. So if you have a mutation, like the variant could cause a mutation in 2 or 3 of them, and it could even get rid of your antibody response. Remember how those antibodies love to bind that one part of the spike protein. Well, if that mutates, well then you lose the effectiveness of all those antibodies. But this means you have 15 different spotlights on the protein. And even it will take 15 mutations to completely get rid of those 15 different fragments. So we talked about 3 examples of cross-reactivity for antibodies. We talked about the antibody to mouse-RAS. We talked about that SARS-CoV-1 and CoV-2 can cross-react. And antibodies raised against SARS spike can recognize SARS-CoV-1 and MERS. This is the cryo-EM one. They probably do recognize common cold spikes. It's just hard for us to see that because everyone already has that when they start. So there's 3 more studies of cross-reactivity, same kind of idea, but with this different system. And that's what I want to focus on this year. For example, this must have made, I went to look into this to see if this has gone along, but it seems like a really good basis for a company that hopefully will work well to make a lot of therapy that will help people. They actually found a receptor, a T-cell receptor sequence that will cross-react with the same peptide in all these different MHCs. That means if anybody has any one of these MHCs, they could just put that receptor into them and it would work for them. So these are 4 different MHC surfaces, and these are fairly common. So basically we would look at your MHC genes, we would say, oh, you've got HLA-B1402. That means that this will work for you. And if you have HLA-C0702, it will also work for you. I can use the same TCR sequence to work for you because it's focused so much on the peptide. And it recognizes this peptide, which is frequently mutated in cancer, I believe. Yes, intracellular oncoproteins. This is really good. I need to look up this company to see if this has really worked for them. But there's a lot of reasons why a great concept still might run into problems in the real world. So this is the most important thing that I knew before COVID started, because I knew about this study. I knew that after being exposed to some viruses that are completely different from other viruses, you will still have T cells that react to completely different viruses that you've never seen. And this is because peptides themselves can be cross-reactive. This is a study that changed my thinking about all this. So here's, if you look at these, they basically had T cells raised to HIV. They had a whole bunch of HIV-reactive T cells. They reacted HIV peptides. And then I don't know what caused them to start doing this, but they started to run those peptides against like A. tropicalis. By the way, if you know what disease that is, but I'm assuming it's a tropical disease, I can probably figure that out. Do you recognize any of these diseases? Flavofascians, B. bifidum. That sounds like something I should know what it is. It's not HIV, that's what I know for sure. All these things up and down, these are even bacteria in some cases. I believe, yeah, L-bacterium, I'm pretty sure that's bacteria. They present these peptides, and you look up and down, and these peptides will react with different clones that were raised against HIV. There's a different pattern for each clone. It has a different preference. But you do have a lot of pluses here, which means that these things are supposed to bind HIV, but they also, as a bonus, bind this thing that the person was definitely never exposed to. Maybe there was some weird thing where a person was exposed to A. tropicalis, but it's definitely not at this scale. This must be something where you have a true case of cross-reactivity, where getting immunity to HIV also gives you immunity to our flavofascians. And we can look at the HIV peptides, we can look at the peptides that it does recognize in the other diseases, and we can say, hey, is there a molecular reason for this? And in many cases we can say, oh, the same types of residues in the middle of the protein are sticking up. So even though most of this doesn't look the same to us, it looks the same to a T-cell receptor. And so the T-cell receptor that recognizes HIV also sees like this phenylalanine right here. And so then it binds it. And there's also a tyrosine right here. And maybe it binds it. Actually, look at this. You can even see what the sequence similarity that might allow this is. PMF, PMY, PMF, PMF. It sure looks like I should expect there to be a PMF in the HIV peptide. I'm not 100% sure because, but you can see that these peptides are similar even though the diseases are very different. And this is one of the things, my kids growing up, kids are always sick, right? Well, part of that is because they are educating their systems, and the idea is even if you have a truly novel pathogen, somebody who's older than the child will have more memory T cells that cross-react. Now, I hope I'm getting it, that this did not save us against COVID. And when I pointed it out before COVID, I was very careful to say, this is a mild effect, it's a T-cell effect, it's going to take some time. It's probably a 30% effect. But it's real, and it will help. So please don't say it's going to be the end of civilization. Let's just work together, and let's try to take care of this as much as we can. And the thing is, if this works for infection, it also means vaccination might provide some cross-reactivity. So flu vaccination, this is one of the other reasons why I think vaccines are a good idea, because sometimes vaccines do bad things that you don't want them to, but sometimes they do good things that you don't expect as well. And as long as, that's why you need safety trials to make sure they're safe, but you can have good things happening, like if you have this peptide, and look at this peptide, there's these other things that are completely different pathogens, but they were found to have similar peptides that were presented. And so they said, hey, let's test this idea. Let's take flu-vaccinated T cells, and let's see if they recognize. And in 3 out of 4 cases, they actually recognized it from 2 different donors. Basically, you can say, if the peptide looks enough the same, then you can get some protection from F. magna. I don't even know what that causes, but the flu vaccine protects against it for these donors that respond to this peptide. The immune system is deliberately messy, and that happens sometimes to have bad effects like inflammation or autoimmunity. But it also can have good effects because once in a while you get something that is a pathogen you've never seen before, but it produces a protein shape like you have seen before, and then you react against that. A lot of people had very off-on visions of immunity, and this is also why people, I think, over-expected the vaccine to save us all, and then to be completely, we should never have a problem again, it should never transmit. Well, it was evolving to be able to transmit. And so that's why when we're looking at the history of COVID, we've got to remember Omicron came along, and it was more transmissive, and the virus was worse in that sense. The good news is it didn't cause as much of a cytokine storm, although still, the jury is still out. If Omicron was the first COVID, would it have been as bad as the alpha wave, as the original strain? So it's that kind of thing. Anyways, I just want you to be able to think about this, and this is another study showing in a COVID context that a different kind of immunization, so MMR and Tdap vaccine antigens will actually produce COVID-reactive T cells. And there is one weird thing, I don't know if this was ever completely chased down, but there's a kind of vaccine they give in Japan called the BCG or the VGC. Can you all correct me? Have you heard of it? It's some kind of bacterium vaccine, and it's just a general vaccine. They notice they give it to your immune system, and you actually have a lot of immunity against a lot of different things. That possibly correlates also with COVID. This is a better study that says MMR and Tdap have a mild correlation where if you are protected against them, you are also protected against some COVID antigens. Very cool stuff. So this is the study if you want to look through it, and you can see some of the information there. But the final example of that is that in May 2020, we had to do a computational project, but I got an idea of I wanted to look for this because I knew that we had similar gene sequences with common colds. How similar could the immune response be? And so this is the module on Canvas. If we wanted to, I could actually give you all this to do again, but we're going to do real research in a lab because we can do that this year. But this is still real research. I mean, you were looking at sequence files, and basically we took all the sequence from top to bottom. This is the entire sequence of SARS-CoV-2 is represented. And what we did is we actually it's not just SARS-CoV-2. What this is actually also the 4 common colds. You see that there's like 6 columns here. Each column is a different virus. From beginning to end, what we did is we took all the peptides. We had the computer say, which peptides will bind MHCs? We took a particular common MHC because we had to just pick one. And then we said, okay, so these peptides, we stacked all the peptides that would bind an MHC from top to bottom. There's a lot of peptides here. This is the actual data. And then what we did is we compared those peptides to see how many of them were similar. When you take peptides from SARS-CoV-2 and you compared it, did you get a similar peptide? And the prediction is SARS-CoV-2 is 60% identical. It takes more than one match, just one bad match will blow up the whole thing. But the idea is that maybe it's a quarter to a half similar. A quarter to a half of the peptides that we have will be similar enough, bind MHC, and present a similar surface to T cells, therefore be cross-reactive. And the big question was, what number should I say is similar enough? If we have a peptide that is 80% similar between SARS and common cold, I predicted from the previous study that that would probably trigger the T cells on average. But if you have one that's only 30% the same, I predicted that probably would not. And the question is, what is the exact threshold you should use? We actually ended up using a fairly stringent one, 80%. And when we had a match within 80%, the peptides were 80% or more similar, it might have been 60%. It was definitely above 50. And I had the students color it green when they saw something, and actually the more similar, the more green it was, I believe. Look at all this green, especially in the C-terminal end of the genome, the 3' end of the genome. Look at there's a lot of hope that there are some antigens from common colds that will give you T cells against SARS-CoV-2. This is how it all overlaps. So these are the 4 different common colds that are coronaviruses. And you have, this is the Venn diagram. There's actually 52 peptides of all the peptides you can make. There's 793 predicted peptides. But almost 10% of them actually overlap with all 4 common colds. This to me seemed to say, well, maybe there's a minority chance that out of the 15 different peptides that you present from being infected from a common cold, maybe you roll the dice, maybe one of them will actually cross-react with SARS-CoV-2. It seemed to me like a pretty good chance that there would be a mild protective response. And so this was just computational, but they recently went back and I based my threshold on the studies that were available at the time. how similar do the proteins need to be for the T cells to be activated against both? And it turns out that in 2022, a paper by my favorite lab in this group came out, and the paper came out showing it was right. Basically, we had picked the right threshold, and our results should hold. And in other ways, our results in general hold. It's still weaker than I would have hoped, but I wanted I would have hoped for a 100% response. Instead, we have maybe a 25% response that you have. You get 25% of the immunity you need. It's a very rough calculation, but there's something there. And so that is the last cross-reactivity study. Which means we have only one part left.
Can I use the restroom real quick?
Oh, yeah.